Hi friends, welcome to my channel Sam Biology Class. Friends, today we will discuss about radial immunodiffusion. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Radial immunodiffusion is a simple quantitative assay that is used to determine the concentration of an antigen in a sample. It is also known as Mancini immunodiffusion. Mancini immunodiffusion. This technique was developed by G. Mancini, after whom it is named, and his colleagues in 1965. Radial immunodiffusion is very simple. In this technique, a sample well is first cut into an agar or agarose gel containing antibody. In the gel, the antibody is uniformly distributed. When the sample containing antigen is placed in the well, antigen diffuses radially outward, forming a concentration gradient. In the gradient, the concentration of antigen is high near the well and low at the margin. In between there is a zone known as the zone of equivalence. We know that in the zone of equivalence, antigen and antibody are in optimal proportions. As a result, maximum immunoprecipitation occurs. In this case, the concentration of antibody remains same throughout the gel. It is only the concentration of antigen that varies. In the zone of equivalence, the concentration of antigen is such that optimal interaction takes place between antigen and antibody. As a result, maximum immunoprecipitation occurs in the zone of equivalence and a precipitation ring is formed around the sample well. The diameter of the precipitation ring is proportional to the concentration of antigen in the sample. In order to determine the concentration of an antigen by this technique, different concentrations of the standard antigen and also the sample are subjected to radial immunodiffusion in one gel. The diameter of the precipitation rings were then determined. In this figure, you can see that as the concentration of standard antigen increase, the diameter of the precipitation rings also increase. From this relationship, it is clear that the diameter of the precipitation ring is proportional to the concentration of the antigen. Based on this relationship, the concentration of an antigen in a sample can be easily determined from a standard calibration curve. The standard curve is plotted by taking the diameters of precipitation rings on x-axis and antigen concentration on y-axis. From such a standard curve, the concentration of antigen in the sample can be easily determined. So friends, this is all about radial immunodiffusion. For more updates, please subscribe my YouTube channel Sam Biology Class.